Yesterday I talked about the variations. I hope you understood the concept. Now let's pass on to the next part of variations. Importance of variations and significance of variations and advantages of variations. Number 1. It makes some individuals better fitted in the struggle for existence. In the struggle for existence. So because of variations only, the animals are able to adapt themselves to the changed environmental conditions. And this concept, according to Darwin, is called survival of the fittest. Survival of the fittest. So the fittest organisms are able to survive and others are eliminated. So this is because of variations. The variations are caused due to changes in the environment. Now as a result, it makes the individuals adapt themselves. Makes the individuals adapt themselves in the changed environment. In the changed environment. In the changed environment. So according to Lamarck and other Lamarckians, so adaptations are universal in nature. Those organisms which are able to adapt themselves to the changed environments can survive. So here, because of variations, the organisms are able to adjust themselves to the changed environmental conditions. Now for the natural selection theory as proposed by Darwin, now these variations provide genetic material. For, that is, evolution, or we can say, specifically speaking, for natural selection. This is another one. So because of changes in the environment, variations occur in the genetic material, and these genetic materials form the raw material for natural selection concept. Now the fourth one, for breeders, those are doing the breeding methods, they are called as breeders. So the variation helps the breeders. What for? To improve the following. Number 1, better yield. In plant breeding or animal breeding, so to improve better yield. Then increased resistance. Then also we have increased what is called the production, that's a better yield, increased resistance. Then we have less input. So better yield, increased resistance, less input. The meaning for less input, the usage of various materials as nutrients for their growth. Then the last one, quicker growth. So the variations help the breeders normally to improve the better yield, increase in the yield, increased resistance against the diseases, then less input, taking only less quantity of nutrients for their growth, and also achieving quicker growth within a short period of time. And finally, the most important one, it is a raw material for evolution. Raw material for evolution. So it contributes the raw material for evolution. Because of variations only, now the evolution occurs. Without variations, there is no evolution. And actually, Darwin stressed the importance of variations, though he didn't know the actual, that is, the meaning of variations, the origin of variations, etc. But he insisted variations are the raw materials for evolution. Such variations are normally caused, we know that one, because of mutation. So these are all some of the importance of variation. Okay, the 5 points you have to remember may be asked in 5 marks question. Let us discuss other things one by one. Let us know few words about the biography of Mendel. What is Mendelism? The various contributions, or simply we can say the contributions of Mendel to genetics is called Mendelism. So it includes the various concepts brought about by Mendel while doing his research work, that is originally in plant hybridization. So these concepts form the basic for the modern genetics. Hence he is aptly called father of genetics. Father of genetics. Now a few words about biography. So he was the first geneticist. Unravel the mystery of genetics or the mystery of heredity. Unravel the mystery. So unravel means open, disclose. Mystery means unknown things, the secret. So he was the first geneticist who unraveled the mystery of heredity. Now he was born in a place in a small village in 1822. So 22nd July 1822 in a small village called Zendorf. This is the name of the village, that is in Silesia. Now this Silesia is nothing but the present Czechoslovakia in Austria. So Zendorf is a village in Silesia, that is Czechoslovakia, and in Austria, the country. He is an Austrian monk. A priest. After completing his school studies, he just reached the Augustine monastery at Rome and then went to Vienna to study mathematics, natural science, that is what is called botany and physics. So he admitted himself at the University of Vienna and studied physics, mathematics and then botany. Then after completing his course there in the university, he returned to one monastery. The name of the monastery, St. Thomas Monastery. So monastery is a place the priests have to undergo the study of theology. To become a priest, they have to undergo theology, the study of religious matter. That is why he admitted himself in St. Thomas Monastery. And he was appointed as a school teacher temporarily in 1849 and continued his interest in plant hybridization. He started his experiments on Gordon pea plant in 1856 and completed in 1863. So this is the duration of the period of what is called experimental analysis. Nearly 7 years he did experiments on, that is Gordon pea plant. For just showing or for understanding the inheritance, he studied the inheritance of 7 pairs of contrasting characters. He crossed and catalogued a number of generations. He crossed and catalogued, that is the experiments for many generations. Then in 1866, he presented his paper and published his paper. He presented and published his paper entitled as Experiments on Hybrids. Experiments on hybrids. So experiments on plant hybridization also you can see. This is the name of the publication presented and published with the title Experiments on Hybrids in 1866. But his analysis was not exposed to the public. So without his experimental analysis exposed to the public, He died in 1884. Whether his publication, whether his experimental analysis was exposed to the public or not. But in 1900, actually, three different peoples rediscovered Mendel's principles. The three peoples who rediscovered Mendel's principles were one. So it was rediscovered only in 1900 by three people. One, Hugo de Vries of Holland. Hugo de Vries of Holland. Number two, Karl Correns. Karl Correns of Germany. Then the third one, Erich von Schellmark. Von Schellmark belongs to Austria. Belongs to Austria. Karl Correns, Germany. Erich von Schellmark, that is Austria. These were the three people who rediscovered the Mendel's principles and exposed to the public. Mendel was the first systematic geneticist in the field of inheritance. What was the reason for Mendel's success? There are so many reasons we have to take as per the book. So, he was the first person applied mathematics and statistical methods actually to understand. So, to just actually apply mathematics and statistical methods. What for? To study biology about the various quantitative data of the outcome of his work, the breeding experiments. So he used mathematics, he applied mathematics and statistical method. Statistical methods to study biology. To study biology that include all the fundamental quantitative data about his experiments. So he also actually studied the laws of probability. Laws of probability. So he applied mathematics, statistical methods to study biology and also applied laws of probability about his experiment to calculate the probability of the genotypes, phenotypes, etc. The second one, he actually studied everything. He provided an accurate data and detailed records that include the quantitative data of his outcome of the experiments. So, he applied scientific methods actually and kept accurate data and detailed records that include the quantitative data of the outcome of his research work. That is the experiments, that what we have, the hybridization experiments. That's about the second one. Scientific methods and kept accurate and detailed records that include the quantitative data of what is called the outcome of his experiments. So his experiments were well planned. His experiments were carefully planned and he used large number of samples. And his experiments were well planned. Actually carefully planned, we can see. The fourth one, the contrasting characters which were taken in Mendel, which were controlled by the genes, the contrasting characters which are controlled by the genes are located on different chromosomes. Genes for contrasting characters are located on different chromosomes. Different chromosomes. The genes for contrasting characters are located on different chromosomes. And that is the main reason to propose the law of independent assortment, we'll see later. Because of the presence of genes located in different chromosomes, he made the law, what is called the law of independent assortment. So he had taken normally 7 pairs of characters. The genes for 7 pairs of characters are located in chromosome number 1, in chromosome number 4, in chromosome number 5 and chromosome number 7. So it took 7 pairs of contrasting characters. The genes for 7 pairs of contrasting characters are located in 1, 4, 5 and 7. In chromosome 1, we have 2 genes, 2 different genes for 2 different characters. In chromosome 4, there are 3 different genes for 3 different casts of characters. And in chromosome 5, 1 gene for 1 character. And chromosome number 7 also have 1 gene for 1 character. So 2 plus 3 plus 1 plus 1. These are all the locations of the genes for 7 pairs of characters what he took for his experimental analysis. We will discuss in detail about the location of the genes later in the lesson. Then, so the parents selected by him for the experimental analysis, the parents selected by him for the experimental analysis are pure breeds. They were pure breeds. I will mention later what do you mean by pure breeds. So he selected the parents which were pure breeds, and he tested the purity of the progeny, and he tested the purity by self-pollinating or self-fertilizing the progeny for many generations, whether they are pure breeds or not. Want to test, he tested, that is actually the pure breeds by crossing, or we can say by doing self-pollination process or self-fertilization process for many generations. Even after many generations, the same character reappeared. So a pure breed is the one which produces the same character for a number of generations when they were crossed. That is the simple definition for pure breeds. We'll have more and more. So that's about actually the pure breeds. Now we'll go for why Mendel has chosen the garden pea plant. The following are the reasons why Mendel had chosen the Rhizophysium sativum for his experimental analysis. Now the plant is annual. The plant is annual. So it shows normally clear contrasting characters. Contrasting characters. Clear contrasting characters. Or distinct contrasting characters or deep contrasting characters. And these contrasting characters are controlled by a single gene separately. Controlled by single gene separately. For example, if you are taking the height of the golden pea plant, height, there are 2 different varieties. One is tall, another one is dwarf. These are all contrasting characters. Tall is opposite to dwarf by taking the character height. If you are taking the shape of the seed, one is round and wrinkled. So the contrasting pairs of characters are nothing but the opposite characters for a particular trait. So we are taking the height of the plant, tall and low. So we have nearly 34 pairs of contrasting characters in this plant. 34 pairs totally. But Mendel took only 7 pairs of contrasting characters for his experimental analysis. 7 pairs of characters. So totally 34 pairs of contrasting characters in that plant. But Mendel took only 7 pairs of contrasting characters for his experimental analysis and crossed the pure breeds individually, separately. That's why he got the results perfectly. Then, see that one, in the case of plant, it's bisexual. Bisexual. That means it's a monoecious plant. Monoecious. Both male and female sex organs are present the same. As it is bisexual, normally in that plant, natural or normal self-pollination took place. Normal self-pollination occurred. Self-pollination. So, Mendel used this plant both for self-pollination or self-fertilization and for cross-pollination or cross-fertilization process, as the plant is bisexual, having both male and female sex organs. Under normal conditions, it is normally self-pollinating plant, self-fertilizing plant. So it is easy for him just to do the process of, that is, self-pollination process and cross-pollination process, or we can say self-fertilization process and cross-fertilization process. Then, the plants are large and also the flowers visible. The flowers are large, so it is useful to study about pollination. It is easy to study pollination and emasculation. As the flower is large, the pollination and emasculation can be done very easily. What is emasculation? The removal of anthers from a flower. Removal of stamens, removal of anthers from a flower. That process called emasculation. So if it is small, you cannot do the process of removal of anther easily, and also you cannot watch the pollination process. But if the flowers are large, it is easy to follow or study the pollination process and emasculation, that is a removal of anthers. So because of these reasons, not only because of these reasons, the plant has only 7 pairs of contrasting characters. Sorry, mistake. 7 pairs of genes, not characters, 7 pairs of genes. That means the diploid organisms actually having 7 pairs of non-genes, once again 7 pairs of chromosomes, you can see. So the diploid number of chromosomes in these plants are 7 pairs. The same number of chromosomes also you can find in the case of wheat plant. There too we have 7 pairs of chromosomes. So in the case of banana, you know that 22 chromosomes. Like that we have in maize or in paddy we have 24 chromosomes. In maize we have 20 chromosomes, like that. So there are 7 pairs of chromosomes. As we have limited number of chromosomes, it is easy to follow, that is, the inheritance of chromosomes along with the genes, to locate the genes in the chromosomes. It is also one of the reasons why Mendel have chosen Gordon pea plant, but this character is not given in the book. Now the next one, Mendelian inheritance or particulate inheritance. Why is it called as particulate inheritance? Because this inheritance establishes the existence of particles or hereditary units or the risk factors, now called as genes. That's why it's also called particulate inheritance. So it establishes the existence of particles, that is minor particles, which are nothing but the hereditary units, also called by factors by Mendel. The word gene was used by Johansen in 1902. But before that, the word factor only used by Mendel. He did use what is called the gene for the factors. That's why it's called as particulate inheritance. So Mendel normally conducted actually a number of experiments. He crossed what is called artificial cross. The artificial cross is nothing but what is called cross-fertilization. So artificial fertilization or artificial pollination, that is also cross-fertilization or cross-pollination. So he did series of artificial or cross-pollination experiments in pure line breeds. Many pure line breeds for analyzing the inheritance. Many pure line breeds are analyzing the inheritance. So what do you mean by a pure breed? Pure breed. So a pure breed is a one that has undergone what we can say continuous self-pollinations. Continuous self-pollinations. That has undergone continuous self-pollinations. Continuous self-pollinations having stable trait inheritance. Stable trait inheritance. from the parents to the progeny, from the parents to the offspring, to the offspring. So, a pure breed is the one, the meaning for that one. An organism which has undergone continuous self-pollinations, having stable trait inheritance. Stable trait inheritance means, for example, if you are taking dwarf variety. So this is what's called self-pollination. Self-pollination. The fusion of gametes from the same individual, same variety. Two dwarf varieties or two tall varieties. This is what is called self-pollination. So when you are crossing dwarf with the dwarf, for a number of generations we are getting only the dwarf variety. There is no new varieties formed. That's why it's called a continuous one, the breeding, and that is called what is known as a pure breed. Similarly, when you are crossing tall versus tall for a number of generations, even after many generations, if you are getting the tall variety, then these talls are called pure breeds. So a pure breed is the one which produces the same one for many generations. That's why I say it includes an organism which has undergone continuous self-pollinations, having stable trait. This is called the stable trait. There is no change, a fixed character. Stable trait inheritance. The same character has been produced for a number of generations, from the parents to the offspring. This is what is called actually pure breed. Suppose mating of two pure breeds. Mating of two pure breeds. So we are making two pure breeds. So mating of two pure breeds that produce offspring. That produce offspring. And that have actually offspring having the same specific parental traits, offspring with specific parental traits. For example, we are talking about tall versus tall. So, the mating of pure breeds results in an offspring, the progeny, with the same specific parental traits, that is tall and tall, the same trait is appeared, and with actually in constant or that are in constant trait inheritance and expression for many generations. For many generations. So we are taking two pure breeds, namely the tall, which result in the production of offspring and progeny. And these progeny have specific parental traits that are in constant trait inheritance. Constant trait means the same stable trait inheritance and the expression of the same character for many generations. So here the constant trait is nothing but the tall character. It is being inherited and it is being expressed for many generations. That is called as a pure breed. So this is one example I am saying that one. Now, actually, pure breed always results in homozygosity. Pure breeds always results in homozygosity. For example, this is a pure breed. We are talking about that one. That is tall versus tall. We'll do what is T for later. I'll express. Now it results in once again tall. So we are getting only homozygous condition in the case of pure breeds. So the pure breeds are always results in homozygosity. There is no formation of heterozygosity. Now, the fusion of male and female gametes from the same individual, that is pollen or ova, derived from the same individual is called, that is self-fertilization. So here for example, we are taking just a pure breed tall and pure breed dwarf. This is what is called actually cross-pollination. In self-pollination, we are taking two tall plants. Now, the gametes. So maybe this is the male plant, this is the female plant, let's assume. So the fusion of male and female gametes derived from the same individual, that is for example in a flower which is bisexual, the union of pollen grains and the egg in the ovule in the same plant is called self-fertilization process. Suppose we are making a cross between two different varieties of the same trait. For example, I am taking Tom versus that is Dove. So it is a cross between two different varieties of the same trait, namely the height. It creates what are called hybrids. So the hybrids forming the first generation. The hybrids are always heterozygous. We'll talk about later in detail. So a cross between purebreds always results in homozygosity. A cross between two different purebreds results always in hybrids or heterozygosity. So Mendel normally studied the behavioral pattern of inheritance and formulated the laws or principles now called Mendelian genetics. So the Mendelian study of behavioral pattern of inheritance or pattern of inheritance and the laws or principles formulated by him together constitute what is called Mendelian genetics. Following are the 7 pairs of contrasting characters studied by Mendel for his experimental analysis, what is called the plant hybridization method. Now, he took the following traits: one seed, another one pod, another one the nature of the flower, anything else related to flowers, the last one related to the stem. So I take now the seeds. Under the seeds I am taking one, the color of the seed. So the color of the seed, now only 2 varieties. One is yellow, another one green. The yellow variety is considered as a dominant trait and the green is considered as a recessive trait. Now about the shape of the seed. Now the seed shape may be round, which is a dominant trait, and another one wrinkled. These two are the contrasting characters. So round shape, that is having smooth surface, and this is a wrinkled one. So for the seeds there are 2 characters. One is the color of the seed, another one the shape of the seed. Under the color, the yellow color cultivated is dominant over the green color seeds. And about the shape, so round shape is dominant over wrinkled character. Now let's go for the parts. The parts normally referring to the dry dehiscent fruit. The dry dehiscent fruit that is observed in the case of garden pea plant, that is called as a pod. The ripened, that is what is called the vegetable. Now again in the parts there are 2 varieties. There are 2 characters I can say. One, the color of the pod, another one shape of the pod. Now the color of the pod, again we have green, and green trait is dominant over yellow. Then the shape, it may be inflated or constricted. The meaning for that one, it's being swollen, it's being normally just to reduce the shape. So regarding the shape, the inflated condition is dominant over the constricted condition. About the flowers, the color of the flower, the purple color is dominant over white. So white is a recessive character and purple color is the dominant character. Regarding the position, we have the flowers found in 2 positions, either at the tip or in the axe. Accordingly, we have axillary flowers and the terminal flowers. Now the axillary flower is dominant. Axillary flower is dominant over terminal. Located at the tip of the branch or at the tip of the stem. Now regarding the stem height, there are 2 varieties. One is tall, which is dominant over another one which is dwarf, which is is a recessive character. So, the diagrams and the pictures of all these pairs of contrasting characters I have inserted in the videos. We can see in the videos about the colorful pictures of these 7 pairs of contrasting characters regarding the seeds, parts, class and stem.